So hello everyone, in this video we will talk about metal ion indicators, which is an important topic under our complexometric titrations. Okay, so in the introductory video of our complexometric titrations I have already discussed a little about how these metal ion indicator exactly work. So the basic thing is that whenever these are present in a complex, they have different color, and whenever they are free, which means out of the complex, they have different color. Okay, this change in color, it marks the endpoint. This is just the basic of our metal ion indicator. Now let us see the definition for it. Okay, so it is a compound whose color changes when it binds to metal ion. So it forms a weak complex with metal ions present in the solution, which has a significantly different color from the form existing outside the complex. Okay, so again the same thing is written, which I explained, that whenever a metal ion forms a complex with the metal ions, so that complex has some different color, and whenever the metal ion indicator is present in a free state, so that is of different color. Okay, this is just the basic of our metal ion indicator. So based on the concentration of metal ions, these change their color, which indicate the endpoint of titration, and these are also called the PM indicators because they are pH independent. Okay, so this was just the basic of our metal ion indicator. Now let us understand that how exactly do they work. Okay, so before the titration is begun in our conical flask, what do we have? We have our metal sample. Okay, we have metal sample along sorry for my diagram. Okay, in our conical flask, what do we have? We have our, again it is like that, we have a metal sample along with our metal ion indicator. Okay, so this metal ion indicator will form a complex with the metal ions in the sample, and that complex will have some different kind of color. Okay, this is our first color or the primary color. Now when the titration is being begun with our titrant, this titrant, which is the chelating agent such as EDTA, it what it will do, this it will displace the metal ion indicator from this complex, and it will itself form a complex with the metal ions. Okay, so here what will happen, metal EDTA complex will be formed, and this metal ion indicator in the complex, it will become free. So when the metal ion indicator will become free, it will result in a change in color, and this will be the second color. Okay, so the change in color from the first to the second will mark the end of the titration. I hope this thing is clear to you, that what is happening. Again I will repeat it, that this is our complex of our metal and the metal ion indicator. So when this is present in a when the metal ion indicator is in a complex with the metal ions, that is having some different color, and when the titration will begin, this indicator will become free because EDTA will itself form a complex with the metal ions. So this free metal ion indicator will have some different color, and that will mark the end of the titration. So I hope this is clear to you. So for example, there is EBT, which stands for Eriochrome black tea, which is one of the most common metal ion indicators which are being used in the complexometric titrations. Okay, so for example, there is any sample which contains magnesium ions, and in that sample we add EBT indicator. So this EBT indicator will form a complex with magnesium ions, and that complex will result in a solution of wine red color. Okay, so now this is before the titration. Now when the titration is being begun with chelating agents such as EDTA, what will happen, this EDTA will replace this EBT indicator from the complex, and itself it will form a complex with the metal ions. Okay, so here metal plus EDTA complex is formed, and our indicator, which is EBT, it will become free. So now when our indicator is out of the complex, it is having some different color, and that color is of blue. So whenever the color of the solution will change from wine red to blue, it means that the titration has been complete. Okay, so this is how the metal ion indicator works, that they result in the change in the color of the solution, which marks the endpoint. Now there are some other examples of metal ion indicators which are commonly used in the complexometric titrations. These are Eriochrome blue black B, xylenol orange, and Eriochrome red. Okay, these are other few examples which are being used, other few examples of metal ion indicators. So I hope you like this video. This is an important topic of our complexometric titrations, and often questions are being asked from this. So thank you so much for watching.